Hello again, and we're going to carry on all this interesting morning with further discussion on verification of research data coming from industry regarding novel smoking products and novel approaches for smoking cessation. We have with us three eminent panelists, Professor Farsalinos, Professor Volti, and I'm not sure if Professor Caruso has the chance to get connected with us. Maybe this will come in a while. And we need to discuss what is our actually work and duty as scientists to check data that come from industry that sometimes, or at least the regulatory authorities, think that they may be some sort of prejudice to support their product. So I would like to start with Professor Farsalinos, who has done a lot of work in confirming in various independent laboratories the several novel smoking products like electronic cigarettes and heat-not-burn cigarettes, and start with him and describe his experience on this field.
Yeah, thank you very much, first of all, for the invitation. There is no doubt that independent verification is a vital part of our understanding of these products, and mainly because of the long but pretty bad history of the industry involving, in many cases, several decades ago, research manipulation, and we understand why this has happened, but we need to learn the lesson, so it's very important to have independent verification of data. Of course, it's something very difficult to do. Regulators are forced basically to accept studies performed by the industry. It's also the only way that regulation can work. The same is happening with the pharma industry, of course, but it's a different story. I think that it's important to the best way that we can, and of course we cannot replicate everything, but we need to make sure that everything that is coming out is well verified, and we need to use new novel tools in that. I mean, we need to ask the industry to present data sets, raw data, for independent scientists to verify. I think this is one additional way of improving research quality and trustworthiness on the data. But I think that it must be a vital part. We all know that science is about verification of results. If something is not replicated, then it's not really science. So in this field, I think replication is crucial, not only because we need to ensure that everything is fully transparent and there is no prejudice, as Professor Ikonomidis mentioned, but also to avoid several mistakes that can happen with these novel products in research methodology, because everyone is experienced with tobacco, cigarettes, and smoking research. This is a very different and sometimes more difficult field of research. So we need to ensure that methodologies and results are well replicated in order to trust them. So that's my introduction, and I hope we're going to continue having a nice discussion with all other members of the panel.
Yeah, I totally agree. Professor Farsalinos has worked also in a clinical setting, but also in experimental setting, trying to confirm or change the results of several industry issues regarding the use of novel products. But let's listen to Professor Volti. He is professor of biochemistry, so I suppose he has a strong opinion on what should we do regarding the safety of the novel smoking products.
Thank you for your kind invitation to the organizer and for your introduction. I totally agree with Professor Farsalinos regarding the importance of the replication studies, and not just for the industry part of the story, but as a scientist, I can tell you how much is important to replicate such studies in order to further understand and to clear demonstrate other people hypothesis or conclusion. So I have a few slides to present regarding the replication, which probably now I will tell you in a few moments the story about my experience with replication. May seems at the first look a very easy task, but it is not like that. Probably it is much more difficult to replicate other people's work rather than starting a new project or an original, let's say, project as we used to do in the basic science lab. So first of all, as you said, I'm professor of biochemistry and I'm chairman of the Center of Excellence for the Harm Reduction at the University of Catania. And together with the Professor Caruso, I am a principal investigator on one of the 8 projects funded in our Center of Excellence that is called the Replica Project, that is seeking to replicate some of the most important work performed by the tobacco industry regarding the harm reduction strategies. So why do we care about replication? So first of all, we have to understand, and that I just learned from myself and with this beautiful experience, understand that replication is not reproduction. So you cannot get exactly the same results as other people are getting in lab. So if you want to measure after exposure with cigarette or with e-cigarette interleukin 6, and for just say they measure 16 picogram per ml, you can forget about you get 16.2 or 16.1. So what you want to do is just replication. So you want to demonstrate that the basic hypothesis is correct. So that harm reduction reduce inflammation as measured by interleukin 6. Doesn't matter if you get exactly the same amount of picogram per ml. It's really hard. There is so much variables that you cannot take into due account when you set an experiment. Then, of course, what we have to replicate, and I think that Professor Farsalino said at the beginning of his talk, we have to replicate only studies that are relevant. So what we decided to do with the Replica project, I don't know if probably some of you had the chance to listen to Professor Caruso talk of yesterday. Of course, we want to replicate studies that have a profound impact on harm reduction strategies. So that's why we choose some of studies rather than others. And then, of course, we want to replicate in a way that we can generalize the original study. So we don't know, of course, we use most of the methods that was used in the study which we are replicating, but we always add additional methods in order to further demonstrate the hypothesis and to convince more about the and make the results more strong. Of course, replication is very important, and the problem with replication, and that is the most difficult part of the of replicating study, is to harmonize the methods, because of course what we do at the COIR is that we replicate in order to make sure about the conclusion. We replicate our studies in a ring trial. So we have 5 different labs all over the world, in particular Italy. We are the coordinating group here at the COIA. Then we have the University of Sultan in Oman, Muscat. We have Serbia, Greece with the Professor Konstantinos Poulas. We have Kazan, University of Kazan in Russia, Indonesia, and we have the Sbarro Institute at the Temple University. So the problem when you want to make a strong conclusion, you have to harmonize the protocols. That it looks simple, but even when you have a very simple technique in the lab, which may be a neutral red uptake, you can just do a simple ELISA test. Then when you want to replicate in 5 different labs, then it becomes a problem. So you want to use the same passage of cells, just making samples. You want always to use the same lot number of the serum that you want to use to culture your cells. You want to use the same kit, the same lot number, the same plastic, the same disposable, of course, all the protocols, same wavelength when using the spectrophotometer lectures. So you have to harmonize, and there is a very nice, even if not very recent work published in Clinical Chemistry that proficiency test most of the hospitals and laboratory has failed because the harmonization was not performed conveniently. And also in science, as I was telling at the beginning of my talk, that is a problem, the replication. There is a very interesting paper came out in May 2020 on Science in the Wire, that the papers that scientists could not replicate were more cited, meaning that we have a problem with I don't want to say that people, they publish fake results, but no science is a perfect science. So even if you can make mistakes in your protocols, of course, most of the study, they are one-center study. I'm talking about basic science. I'm not talking about clinical trials. Most of them are multicentric. So you are sure about your results you just published, but if nobody is replicating them or is able to replicate them, then there is no strong conclusion that science, and we cannot assume that as a correct data. So we are starting from this principle that no science is a perfect science, and if some study has to impact profoundly on public health, such as harm reduction, then we have the commitment as a medical doctor, as a scientist, to replicate that studies, to offer to our patients the best science possible and the right information regarding their personal choice of switching to a normal cigarette, to an electronic cigarette, or to harm reduction strategies. Of course, as we know, the best way is always quit smoking, but we know all the story. I don't want to get in, but we have to provide them with solid scientific data to make them choice scientifically correct. So thank you for your attention. So that is my point of view. If you have any question regarding our experience with replication, I would be nice to answer your questions.
Thank you, Professor Volti. And as we have Professor Caruso with
Sorry,
us.
Mr. Chairman, I am Lee Volti. That's always because there is a space between Lee and Volti, so it's always my fault at the beginning of every foreign session. I'm not Chinese, so I'm not Lee. So Li, so I am Lee Volti. It's like
Professor Lee Volti.
So it was my fault.
Okay, sorry for the wrong spelling. And let's go ahead and ask Professor Caruso. He is also professor of biochemistry, share his views with us, and we can discuss all in the middle and have a second round as well. Professor Caruso.
Yes. I have not the presentation today because I showed yesterday, and I showed the experience of the first year of replica. As Professor Volti says before, it was a very amazing and difficult project because of the harmonization. We have to understand that we are working in this period with the standards created for cigarette smoke. So our first duty was to say if the methods used are good for tobacco smoke and electronic cigarette smoke. So, for example, we performed an experience with the cytotoxic test in order to evaluate if different cytotoxic test or viability test can be used preferably with one of these products, and we published this preliminary results recently. And for example, we observed that effectively the MTT is not a good test for cigarette smoke. In fact, the neutral red uptake assay is the preferred test for cytotoxicity that is indicated by guidelines from FDA and other regulatory bodies. And another point that we raised was the comparison of smoke and vapor in order of number of puff released from the product to the cells. And we conducted another preliminary research on nicotine released and stability of nicotine in the media and in the Cambridge filter pads in order to receive from our partners around the world the samples and quantify the nicotine in order to, first of all, establish the comparability between the smoke and the vapor that we use to expose the cells, and in order to compare the ability of each lab involved in the study to use these machines, these products. In particular, for cigarette, it is really difficult to obtain the same exposure for cells because the cigarettes are products not really standardized, but with certain limits. And so we spent 1 year, I think, in order to do this preliminary studies in order to obtain a certain harmonization between all the labs. So there are difficult studies. Sorry for my telephone. There are difficult studies that we are trying to optimize year by year with our partners. And this kind of collaboration with different laboratories around the world, I think, is a great option. Sorry, another time. A great option for our future because data obtained. Sorry, just a moment.
So I just would like to carry on until Professor Caruso comes back. I realize that there might be different methods to measure, for example, nicotine release, or to measure the cytotoxic effects, or to measure even the percentage of aldehydes or other toxicants. But are industry data and independent data on the same trend, or is there a difference and one conflicts the other? What is the situation today? Maybe Professor Volti could answer that, and then Professor Farsalinos to this question?
Of course, you are completely right. Of course, industry, they have a lab in their companies as we have at the university. There is no one method better than the other. I always say that two methods different methods giving the same results is better than one. So that was what Massimo was trying to say before. Let's say just the example of Nitro Red. It's a very reliable test, but is also based on the intracellular pH, because it's based on the uptake of fluorescent assay. But when you release nicotine, of course, you are changing the pH of the cell, then that could represent a bias. So what we wanted to do, of course, this is the gold standard to measure for the cytotoxicity assay, is an excellent method. But when you use nicotine, then we was wondering, using the right method, maybe if you use a different method, we are underestimating or overestimating the toxicity of the e-cig or the normal cigarette. So that's why I was saying in my talk that we decided to generalize our conclusion, to add the different methods. So in our case, we use a simple MTT, which is based on mitochondrial polarization. So it's nothing to do with the uptake of a fluorescent dye, and we also adding a free label assay, which is based of microcurrent measurement on a free label assay. So they all gave the same general results, of course, confirming the starting hypothesis of the paper we decided to replicate, but to different extent. So maybe neutral red, it's okay, but then we have to remember that since the pH is variable because of the nicotine, we are trying to overestimating the cell toxicity with the nicotine-containing products. So
I realize that, of course, if you use a different method, you are not going to get exactly the same measurement, the same value of a certain marker. However, the trend should be on the same side. For example, when you measure increase, all methods should show an increase. When you measure with one method decrease, then all methods should show a decrease. Of course, there may be difference in the percentage of increase or percentage of decrease, but at least it is confirmatory that an intervention, or I don't know, a cytotoxic intervention goes to the same line, to the same trend.
Exactly. I give you another example. Apoptotic cells, when you expose the cells to smoke or to e-cigarette vapors, of course, some of the cells, they go to apoptosis, but when you use Nitro Red, then apoptotic cells are still able to uptake the neutral red dye. But if you do a cytophotometric analysis with the Nexin 5, then you are able to exclude the apoptotic cells from cell viability measurement. Then my opinion is like to measure cytotoxicity is better to use Nexin 5 Yodur propide methods rather than, of course, these needs to have a cytophotometer inside the lab. Then it means that you have a specialized personnel to use that particular machine. But the neutral red give, in terms of trend, as you were correctly saying, the same results of the cytophotometric analysis.
Okay, Professor Caruso, would you like, because you were interrupted before, to sum up, or?
Sorry for the distraction. But yes, I heard that Professor Volti completed my thought, and I would only highlight that we are trying to replicate studies, but to confirm any details of the cytotoxicity tests in the specific case of electronic cigarette and tobacco heating products, because I think that we need to confirm this kind of protocol, as for example Coresta is doing, is a priority for us.
Can I ask you, is there any cytotoxic effect of the new products at the very end? Because this is what interests us. Okay, there might be difference with various techniques, but is there an effect?
From our studies, we observed a very slight effect of cytotoxicity, and the reduction of 90% is strongly confirmed by our results. But at the moment, we are working on acute exposure. We are planning to extend the time of exposure in order to evaluate other aspects of chronic or 15 days exposures for cells or for any kind of models.
Okay. And Professor Farsalinos, your experience on that?
Yeah, I fully agree with both Professor Volti and Professor Caruso that there is a big problem with standardization and harmonization of not only procedures, and it's not only about the methodology of the experiment itself, it's also the problem of collecting the aerosol in many cases. For example, with electronic cigarettes, you know, we don't have one device or one set of parameters that works in all devices. It's basically the number of different combinations of equipment, battery, power settings, atomizers, and liquids is immeasurable, basically. You know, it's you cannot even imagine how many combinations there are. And every different device works differently. So there is a very big problem, and it's a real problem of standardizing even the collection methods. Coresta is one organization of several scientists, mostly of the industry, who are working on standardizing the methods, but still you have other things to consider when you are collecting aerosol, like, for example, the puff duration and the power settings, which are very different between different devices. And we have seen, since you asked previously about our experience on whether we have verified or rejected previous findings, I have rejected several findings, not from the industry, but from research institutions, universities, mainly in the US, which was due to bad function or basically misuse of the equipment. So they basically abused, and that's one of the commonest things that we are witnessing with electronic cigarettes, because that's an aspect of electronic cigarettes. It's the abuse of the power settings mainly, which result in conditions that are basically impossible for anyone to use. You know, it's like wanting to see if bread contains any carcinogens. You put it in the oven and you then literally create a piece of charcoal by heating it at 300 degrees Celsius for 3 hours. You know, this is a piece of charcoal. This is going to be full of carcinogens, but it doesn't mean that bread contains carcinogens, and it doesn't mean that this piece of charcoal that you have produced is edible. No one is going to eat it. So this is an issue that I've experienced, I think, since 2015 with electronic cigarettes, and it needed a lot of work in order for people to understand that, you know, it's very easy to get a high-power device and literally burn everything. And of course, when you burn everything, the end result, the aerosol that will be produced, is going to be a disaster in terms of contaminants and toxic compounds. But you're creating conditions that are not realistic and are never going to be used by anyone. So, and you know, for people who have been used to testing and analyzing tobacco cigarette smoke, it's very hard to adjust to the new technologies, which are very different in the way they are used and in the easiness with which they can be abused. So these are practical issues that I think still have not been resolved. I think that Professor Caruso and Professor Volti are doing a great job. It's basically their job to find ways to standardize, methods. But I think at the end, for some cases, and I'm talking mainly for electronic cigarettes, it's going to be very difficult to standardize everything. So this is creating also practical issues concerning the verification process and of research.
Can I ask all the panel, what about the heat not burn products that are a little bit more standardized in terms of nicotine production and emission of the gases? I realize that, of course, electronic cigarette, there are so many different devices, there are different liquids, different use, time of use, and all these issues. But what about the heat not burn cigarettes? Is it easier to do verification of the industry results?
In short, I will tell you my experience since I've done independent studies on one of the heated tobacco products, and you are right, the procedure is much simpler. I've done a couple of studies evaluating both toxins and nicotine delivery, and I can tell you that I have fully verified basically the industry results. And in cases like that, chemical analysis, which is very easy for everyone to do, I think that the industry cannot hide, and it would be pretty unwise for them to hide things because it's much easier for such studies to be replicated. So I hope that they are honest. We've seen that in the studies I've done, and I fully agree with you that with these products, it's much easier to standardize procedures.
Professor Volti?
I totally agree with Konstantinos Farsalinos. The only thing that we realized with tobacco heat products was always to use new products in our testing, because if you try to use and recharge and to reuse many times the same products, then you may not get reliable results between one session and to the other. So of course there is always the same trend as I was saying you before, but you may get in terms of quantitative analysis different results. So what we do is always to use new products and to reuse a few times, not to exhaust too many times the same device. And if may add something to what Konstantinos said before, it's of course a matter of watt and power when we test electronic device, but also it is important the way we test the vapor on, of course, for the in vitro studies. So I am getting sick of looking and reading papers that are exposing bronchial epithelial cells with some media in which was bubble the smoke. That is not the way of exposing bronchial epithelial cells to smoke, because in our airway tract, the epithelial cells, they should be exposed to air liquid interface, because they get directly in contact with smoke. So there is no way that there is some water in which we bubbled some cigarette or electronic device. Then of course we are getting not clinical relevant results. So that is a very important way when we look at papers and reading papers from the others, to make sure that they use the right way of doing the experiments.
Okay, the Professor Caruso?
Yes, I agree too with Professor Farsalinos and Professor Volti. And in fact, normally we prefer to test closed systems, for example, e-cigarettes with pods, because is more fairly to report the results on this kind of products, because in the variable products, we can, for example, I am writing a paper on conducted with this kind of product, but I'm reporting the number of holes for air opened during the experiment, and I'm maintaining constant this number of holes for the entering of air during the vaping. So as Professor Farsalinos said, the variables in this kind of devices is very, very great. And many papers are conducted with exposure very ineffective. In fact, when we expose bronchial cells to conditioned media, we lost different substances that are not hydrosoluble, and is not. So I agree completely with my colleagues, and normally when it is when I receive a manuscript to be revised, is the first thing that I look if the exposure is conducted in the correct way, in an acceptable way.
Yeah, I suppose it is very difficult to standardize the amount of vapor, and that's why you say how many holes has the equipment that releases the vapor from a single heat stick, for example. And
I wanted to make a comment. I fully agree about the need to do in vitro studies that have some clinical relevance. And I fully agree with both Professor Caruso and Professor Volti that you need an air interface. But you know, this is also a much more expensive methodology. So I think that other methodologies which may not be, you know, that relevant clinically may be helpful when you are just using them by comparing different products. So they can be used not to create clinically meaningful results, but to compare tobacco cigarettes, for example, with all these products. Now, the issue that comes out is how are you going to do a comparative assessment? So how are you going to compare two things at the same quantity? And I think that in that case, I suppose we can all agree that using nicotine as the unit of exposure is one pretty reasonable step and solution, because we know that everyone who is using nicotine products, they use it in order to obtain the nicotine they need. Usually everyone adjusts their patterns of use when they're using different products, because at the end, their nicotine levels are basically identical, which means that their total nicotine intake is the same. And I think that nicotine can be used as a very valid and easy to measure comparator of exposure. But at the same time, for clinically meaningful results, there is still a problem of how much of the product you are going to use, of the aerosol or whatever, to expose the cells. And how can you prove that this is meaningful in a clinical context in human use? So there are issues that need to be resolved, haven't been resolved so far. So we're still working on these methodologies. I think it's crucial to start with a methodology. Otherwise, you know, we may have a lot of results that we don't know what to do with them. So yeah, fully agree with everyone else.
Okay, but do we have positive or negative results regarding cytotoxicity, for example, for the heat-not-burn products?
Well,
what is the situation up to now from your experience, from your laboratory work?
I think that I will agree with Professor Caruso that the results are showing vast reduction in cytotoxicity from all these products, both e-cigarettes and heated tobacco products compared to tobacco cigarettes. In many cases, we don't see any cytotoxic effect. In some cases, we see mild cytotoxic effects, which may not be clinically relevant, but what I'm referring to now is using the tobacco cigarette as a comparator in all these cases, and of course, measuring similar nicotine exposure with all these products. So in all these cases, I think, you know, it's going to be extremely difficult to even compare these products. I mean, that's not very scientific, but I'll tell you the experience of collecting aerosol and smoke is also extremely impressive in a laboratory. When you are bubbling and trying to
I may add what is impressive also when you look, not just the smells, of course, especially for those like me, they don't smoke anything. That is really bad when we have to do the session. But also when you look at the filter pad that we put behind the cigarette or the THP or the electronic cigarette, when you look at the cigarette, it's completely yellow. When you look at the THP or the e-cig, it's perfectly white. So just looking at that, you get what people smoking they put in their lungs. I think if you put, I like to put some of the smokers of my lab, they smoke a lot, and put it in to see what we do the session, to see what you put in your lungs. And I show them the yellow, so they start switching to that. So that was a good lesson of harm reduction. So now they don't smoke and they switched to a harm reduced product. So maybe
Professor Caruso,
all of them in the lab.
any comment?
At the time, I absolutely agree with Professor Farsalinos. We are facing problems in order to eliminate contaminants from glass after different times that we cleaned with detergents, and we decided to dedicate an exposure chamber only for air, an exposure chamber only for smoke, and so on, and with the impinger for the bubbling of smoke and vapor is the same thing. And it is impressive, the contaminants that are released by smoke, and that is difficult to eliminate from the laboratory instruments. And this is the reason, because we have a smoking machine and a vaping machine. It's impressive.
Okay, can I ask you, I gather that all of you agree that there is less cytotoxic effects either from electronic cigarettes or from heated non-tobacco products compared to combustible tobacco smoking. Are there any sort of data or results that are borderline and you would like to reconfirm? That's one question. The second question is, which one of your experiments you would like us clinicians to test in the clinical setting? And this is for all the panel. Let's start from Professor Volti.
Personally, the results we're getting, at least in vitro, they are really, really clear. So we have no doubt about the fact that these products are harm-reduced products. So we are talking about 95% reduction in toxicity with e-cig or THP. So the results, it's nobody, I think, can say anything regarding the robustness of the results. Probably the results we are doing some pilot studies. I would be fascinating regarding the effect of these products on behavior, so on central nervous system. Then that would be probably something that at least we cannot provide in vitro, and I would like to see some of the results we are getting in vitro in a clinical setting on the central nervous system regarding addiction, regarding aging. That would be interesting, but maybe it would take too long to see some of these results, because e-cigarette THP are so less toxic than cigarette that maybe we have to wait for 20-25 years to see some of these interesting results.
Professor Caruso? If you could unmute your microphone.
Sorry. Yes, I agree with Professor Volti, but I would like to. We conducted then studies on people with asthma in order to understand the effect of smoke on these pathologies, asthma and COPD. But it would be interesting to test our results on, for example, a cohort of naive vapers and without any kind of pathologies. But in Italy, it's difficult to conduct this kind of study with bronchoscopy and see bronchial brushing and so on, because we are focused in this period on bronchial epithelial cells. I think that this could be the first result that I would see confirmed in a clinical settings, in a clinical setting.
Maybe, for example, you could check the release of nitric oxide. This can be measured and see the effects on the release of nitric oxide from trachea and the bronchial tree. This is another method that they use it in some cases that they want to show the functionality of bronchial cells.
Yes, we observed this parameter, but this parameter is strange because is reduced on smokers and is increased in asthmatic people, for example. And so it's an indirect parameter. It will be really interesting to see effectively the cells. But I participated in a study with Bretomics, that is an omics technologies that analyze volatile organic compounds from breath, and we are thinking to this kind of project for the future in subjects that could be interesting. But normally I like very much to work on cells, so it will be very impressive to have a direct observation of cells from smokers, vapers, and no-smokers.
And Professor Farsalinos?
Yes, I think that in the clinical setting, we need to be more clever. The problem with smoke is that we know pretty well that smoking causes disease over a long term. And in order to even see improvements, reduction in risk, you need to wait for years, especially when you're looking at healthy people who are smoking. So what can you do? You need to shorten the time. This can be done in two ways. First of all, work on people with already established smoking-related disease, because these people, when they continue to smoke, they have a more rapid progression of their disease condition, or in the case, for example, of coronary heart disease, they have a higher risk of developing a second heart attack pretty rapidly within the next 1 to 2 years. So looking at people with disease will give you an idea of what's happening when you switch from smoking to these products compared to continuous smoking. And the other way of working with that is trying to find markers of disease, so prognostic indicators of disease, or other smaller, maybe markers that are not clinically meaningful today. But for example, we know that smoking is a chronic carbon monoxide poisoning, for example, and we know that vapors experience an improvement in their exercise capacity. So maybe we should work on markers like that, measure exercise capacity, measure VO2 max, doing such measurements which have not been done clinically, and it is meaningful work. Of course, working with huge cohorts and waiting until some healthy people who were smokers until now will develop disease as continuous smokers or as switchers to other products, we're talking about decades. It's basically impossible to generate any meaningful data now. So we need to approach it in a more clever way and find ways that will provide prognostic information for all these people.
Okay, just to sum up, as time flies,
Professor,
it was a very fruitful conversation.
Professor,
Okay.
sorry, I
Yes.
see one question in the chat. I would like to answer if you give me a few minutes.
Okay.
I see one question in the chat by Mr. Richty, RTS Media, regarding the fact that the COIA received millions of dollars at Philip Morris Foundation, and how we can say that our studies are independent. So since this is a question that we get asked frequently, I would like to make this point clear once for not going back to this point for the future. So what we did at the COIA, we just submitted a grant to the Foundation of Free Smoke World, which received a grant from Philip Morris. Our study is completely independent since I am a full professor of biochemistry, tenure track, so my stipend is completely paid by the university. So I just get the money to replicate the study. I decided to replicate it together with my 5 partners all over the world. Nobody has to question regarding the protocol I wanted to use to replicate these studies. Nobody is going to read my paper before submission and always declare my conflict, the potential conflict of interest in the papers I published. So I don't understand why I keep asking this question, since this study is completely independent, since we have to publish just what we get in our results. Our data are open source, so anybody from all over the world can investigate and replicate and decide to see what we did in our study. So on the other side, I don't understand why other people, they get paid salary from some probably agency, and they, without any scientific data, keeping asking these kind of questions. So probably they should declare their conflict of interest before asking such questions.
Okay, thank you very much for your comment. The point is well taken. I'm afraid we have just concluded our time. And just a last minute conclusion, I think all of you agree that we need harmonization to the methodology used in the laboratories, and this can be found within research among the various laboratories, and probably a cooperation and a consensus paper documenting which are the most appropriate methods to check these products in the lab. The second is that all methods, although different in their extent, they show a similar trend that these products show beneficial effect in terms of toxicants, in terms of cytotoxicity. And of course, the last conclusion is that we would like to see these results replicated and translated, actually, because it's translational science, from the lab to the clinical setting. And Professor Farsalinos offered solutions. I agree with him. I have used surrogate markers because otherwise we will have to wait for another 10, 20 years to see major cardiovascular events occurring and compare the various products. So we can use surrogate markers of lung function, vascular function, cardiac function, and see the effects in comparison to the tobacco combustible products. And with these few lines, I would like to thank you for this very stimulating discussion. And I would like to call for the next very interesting session discussing about smoking and COVID with eminent chairman and eminent speakers on this subject. Thank you very much all.